Hey everybody, I want to take a quick minute to walk you through the last two pages of the assignment from class on Tuesday, so nucleic acid structure. So I'm not going to make this as a homework, but rather I'm going to say, hey, or the pre-assignment and say, hey, do it on your own time. And then if you want to check your answers, you can watch this video, which I highly recommend that you do. All right, so the last exercises are about the Tata box, which is again a sequence of DNA that's TA rich. And it's where these TA rich sequences come ahead of the promoter for a gene. So the Tata box protein binds the Tata box sequence, it pulls down the polymerase onto the promoter, and then it turns a particular gene into mRNA, into RNA, which is then goes to the ribosome and is turned into proteins. Now, so we're thinking specifically about the Tata box. And the first question is about small molecule drugs, one of which is distomycin. These molecules are known to bind to DNA. And so the question was, based on this picture, try to figure out which groove distomycin is binding to. It's a little bit hard to see in this picture. You can never really get long enough DNA to see it well. But distomycin is binding to the minor groove. So that's the part where the backbones are closer together. And the major groove would be up here. Again, you can't see it so well because it's partially cut off, but on this segment of the DNA up at the top, the backbones are further away from each other, so that would be the major groove. Now, small molecules like distomycin are known to be minor groove binders, and they have these little rings. So these are imidazole rings, but sometimes they also contain, or modified imidazole rings, sometimes they can contain paroles. And so in theory, you can switch up the identity of those rings to target different sequences of DNA. By the way, these molecules are called polyamides because they contain multiple amide bonds. And they prefer AT-rich DNA. Now, the reason why I wanted you to get practice looking at a small molecule is because I wanted you to look at and identify where the Tata box binding protein does. So again, we're learning more about the Tata box binding protein. And here is the crystal structure of the Tata box protein binding to a DNA strand in space. Now, it's easier to identify the major and minor grooves, which are drawn out in this structure. However, it's a little bit hard to figure out where exactly the Tata box is. And so you'll have to take it for a word from me that the Tata box binding protein is binding to the minor groove of DNA in this case. Now, if this doesn't surprise you, this should, because it's typical that small molecules like distomycin tend to bind to the minor groove of DNA, whereas large proteins that come in and regulate or mess with the DNA very often bind to the major groove. It makes sense. A protein is a bigger molecule that would bind to the major groove. However, the Tata box binding protein is an exception, and it actually binds to the minor groove rather than the major groove of DNA. The vast majority of other DNA binding proteins you read about will bind to the major groove. So now we're tying this in together with the sequence of the Tata box to what we've talked about in class. And so I asked you to see what you noticed about the Tata box sequence. So if we scroll up to the intro to the first question, the Tata box sequence is CTA-TAAA. Now, the Tata comes from TATA, but there's two things that you should notice. The first thing you should notice is that it's AT rich, and the second of all is that part of it is alternating T's and A's. So why is this important? Well, if we think about it, if we looked back at our exercise where we ranked the relative stability of the base stacking interactions between DNA base pairs, the weakest, the sequences that had A's and T's were weakest, and that had partially to do with the base stacking and partially to do with the number of hydrogen bonds. But all AAATTT was weaker than sequences that had G's and C's in them. However, alternating A's and T's are less stable than straight A's and T's. So if we look at the sequence of the Tata box, except for the C at the beginning, it's all T's and A's, and in fact, we're alternating at the beginning of the sequence. So What this suggests is that the Tata box sequence has a really low, would have a relatively low TM, or that melting point transition. But this is a good thing, because if you think about what's happening at the promoter, or the part before the promoter of the gene, the proteins need to insert themselves into the DNA so they can spread apart the bases so polymerase can get in there and do its job and transcribe DNA into RNA. So sequences that need to be pulled apart easily in DNA, like at promoters, so where transcription needs to get a head start, are often TA rich and often even have alternating T's. So if you were to want a sequence that you wouldn't want pried open, you would want it to be CNG heavy. But in this case, we want the DNA to be easily melted by the Tata box binding protein and other accessory proteins, so we would want to have an AT rich sequence. Now, part D is tying this back to thinking about protein structure. And so it's saying, okay, so DNA has the three parts. It has the phosphate backbone, it has the ribose sugar, and it has the bases. And so the protein, the Tata box binding protein, in order to bind the DNA and then pry it open, needs to have some sets of different amino acids that can interact with different parts of the protein. So in particular, it needs to have amino acids that can work with the backbone, the phosphate backbone, and then it needs to have amino acids that interact with the bases in the center of the DNA. And if you'll remember, we talked about how although the DNA bases have heteroatoms, so N's and O's, they're also hydrophobic because they are greasy and aromatic and flat planar rings. So if we want for the amino acids that we want to interact with the phosphate backbone, we would want them to be positively charged so they could neutralize the negative charge on the backbone. So we would expect these amino acids to be amino acids like lysine and arginine. And then for the amino acids that we would want to go between the bases, we would want amino acids that could pi stack or form hydrophobic interactions with the bases. And so good guesses for these would be phenylalanine, tyrosine, or tryptophan. And then lastly, we've set this up to say, okay, we talked about small molecules, but the reason why I brought up small molecules here is because small molecules are a promising class of potential drugs. So one of the problems that happens with diseases like cancer is genes get over-transcribed in cancer cells. So specifically, genes that are related to cell growth and development oftentimes are transcribed out of control. So one of the levels at which we can prevent overexpression of the wrong genes is by cutting them off at the DNA level. And so in particular, could we possibly prevent Tata box binding protein from being able to bind to the Tata box or other AT-rich sequences of DNA? And so the theory behind small molecules like distamycin as drugs, so distamycin itself would never have any potential because it's way too nonspecific. But if we could devise a molecule like distamycin that could specifically bind, we could synthesize it so it would prefer the Tata box over other sequences. In theory, these molecules could compete with the Tata box binding protein to bind the Tata box sequence. And so if you can get the small molecule to bind to the Tata box instead of PDP, then you could control whether or not genes were being transcribed when they should be. So if you see someone that has cancer symptoms, you have certain proteins being synthesized out of control. If you could synthesize a molecule that would bind to the gene promoter instead of the Tata box protein, in theory, you could shut down the production of some of those cancer-associated